A very good evening for one and all. Our CellTalk presenter for today's session is Nupur. So Nupur Biswas is working as a senior bioinformatics scientist at Renix. She's working on bioinformatics and other multiple projects as well. Some of her projects include designing personalized neoantigen vaccines for cancer patients and exploring the genes behind carcinoma and the health applications of digital healthcare wearables. Nupur has a doctoral degree in physics and her research work focused on studies of confined polymers. Before joining Renix, she worked at the Indian Institute of Science, Raman Research Institute, Kolbatri, and the Indian Institute of Chemical Biology. At Renix, she has gained experience as not only as a research mentor, but also as a scientist, contributing to various international publications. So, over to you, Nupur.
Thank you, Bharati, for your nice introduction, and welcome everybody in my today's presentation. Today, I am going to talk about a paper which is titled Lung Adenocarcinoma Promotion by Air Pollutants. This paper was written by multiple authors, and William Hill is the first author of the paper. It was published in the journal Nature in the month of April of this year. Although this research article is primarily focused on the lung cancer, but it has a broad impact on other pulmonary diseases. Now, coming to the article. This article is concerned about the role of the pollutants in case of lung cancer. We all are aware of the increasing pollution in our surroundings. Our atmosphere is contaminated by a wide variant of pollutants. There are multiple sources of pollution. Outdoor air pollution includes the particulate matters emitted from the vehicles, chemicals from the various industries, and it also includes microorganisms. In the case of indoor air pollution, there is a main source of indoor air pollution is the tobacco smoke. And also another source of major indoor air pollution is the cooking fumes, which is particularly concerned in the village areas. Now, who considers this pollution as a major cause of several pulmonary diseases? It's because this pollution has multiple effects in our body. They induces oxidative stress, inflammation, cause DNA damage, induce mutations in the DNA, changes the DNA, induce DNA methylation, modify the RNA, and it also affects the metabolism. Now, coming to the lung cancer, we know that smoking is a major cause of lung cancer, but it is observed that lung cancer in never smokers is also a big issue. It is the 8th most common cause of cancer death in the UK. So, this article is focused primarily on the lung cancer in never smokers. Such patients bear distinct clinical and molecular characteristics compared with lung cancer in smokers. And these molecular characteristics include EGFR mutations. On the other hand, the polluted air contains different types of particles. That is, here we are going to discuss about the particle matter whose size is less than 2.5 micron. These tiny particles can travel deep into the lungs, which is actually a barrier organ of our body. And these particles are further responsible for heart diseases and lung cancer. Now, traditionally, it is thought, or it was observed also, that carcinogens cause tumors by directly inducing DNA damage, by inducing DNA mutations. And pollutant, these mutated molecules cause sequential accumulation of mutations, which often leads to cancer. However, recent data suggests that there are some carcinogens which may not cause detectable DNA mutational signature in tumors. So, but they are also responsible for cancer. And also there exists wide geographical variance in the occurrence of cancers. These things are need to be discussed. Coming to the EGFR gene. EGFR is the epidermal growth factor receptor gene. It has very important role in our body as it triggers several signaling pathways. Its mutations and amplifications are also reported in cancer, and strong association is also observed. Mutations can happen in multiple sites of locations of EGFR. However, common mutations are deletion of exon 19 and the point mutation L858R that lies into arginine at the location 858. This manuscript is primarily focused on this point mutation. And there is another issue, that is the smoking status of the patients. EGFR mutation, effect of the mutation depends on the smoking status of the patients also. With this background, the challenge remains to control the spread of the disease, definitely. But controlling pollution is, of course, the best solution, but we know that it has multi-layered challenges across the globe. And also, we need better understanding of the effect of pollutants on the genetic diseases and how it can be prevented. With these challenges, the objective of this current research is set to explore the relationship between PM2.5 and EGFR-driven mutations in the context of lung adenocarcinoma. It presents a multidisciplinary analytical and technical tool using human lung samples and mouse lung samples also. For this research, researchers has used multiple databases. For the England population, they use data from PEACE, TracerX, UK Biobank, and DRS databases. For the Asian population, they have taken data from South Korea and Taiwan. This research used multiple experimental techniques, including digital droplet PCR, MiSeq, duplex-seq, whole genome sequencing, RNA sequencing. One important part of this research work is the use of the mice models. They used genetically engineered mouse models suffering lung cancer, lung adenocarcinoma, LUAD. And they induced expression of oncogenic human EGFR L58 mutation in the mouse lung. And then they studied the mouse in presence of particle PM25 or some control, so like PBS buffer. The exposure was continued for 3 weeks, and the study was checked, and the effect was checked at the end of 10th week. Now, first, they wanted to find the correlation between the lung cancer incidence and PM25, whether these two parameters are really associated. They observed that lung cancer incidence increases with PM2.5 levels for all 3 countries, England, South Korea, and Taiwan. And we observed that the increase is more in case of the Asian countries. The curve is more steep here, slope is higher. That means it is more prominent in the Asian countries. This association is more prominent in the Asian countries. It may be due to the fact of the level of the pollution. We see the level of pollution is quite high in case of the Asian countries compared to the England. Then they further checked the association of other variables with the lung cancer incidence. These variables include the smoking status, age, sex of the patients, even the education of the patients. And they check that smoking has a high smoking, and as well as the particle exposure to PM2.5 has more correlation, and that means they enhance the risk of lung cancer. And for this study, they ensured that the participants who have lived at the same location for 3 years are considered. Then they checked the lung cancer incidence and EGFR mutations. They observed that researchers in case of the lung adenocarcinoma, specifically, not all types of lung cancers. They found that the frequency of EGFR-driven lung cancer cases was significantly higher after 3 years of high air pollutant exposure. It is increasing with the occurrence of the EGFR-mutated lung adenocarcinoma is increasing with the increased level of particle exposure. And they also studied that this association of PM25 exposure and lung cancer adenocarcinoma occurrence is quite strong when the exposure time is 3 years, but not so strong for 20 years. Here we see 72% patients are having the EGFR mutations when they are highly polluted, when they are living in the high polluted area. But in case of the 20 years exposure, it reduces to 50%. It may be the reason that 3 years of exposure is enough for the EGFR-driven lung cancers to arise. Then they studied the mice model to correlate how the genetics is influenced. For that, they took the mice suffering in the LUAD adenocarcinoma, then they injected the induced expression of the mutated EGFR in it, and then exposed to the polluted environment, to the cells, and then check the immunoprofiling, organoid formation, and whole genome sequencing in this. The result shows that particle exposure of particles is promoting the lung cancer. When the 50 microgram particulates were exposed to it, there exists an increase in the number of pre-invasive neoplasias. And here it shows the dose-dependent increase. Increase is more for the 50 microgram exposure compared to the 5 microgram exposure. Here also the same thing is illustrated. These cluster sizes actually represent the number of EGFR mutated cells. So there exists a correlation between the dose-dependent increase of the number of neoplasias. Yes. Now, to explore how this is happening, they did the spatial analysis of clonal dynamics, and it revealed two facts. One is the number of cells increase into the cells grew into the cluster, and also within a cluster the number of cells increased. That means that if the exposure to particulate increases the number of EGFR mutant cells and also the proliferation rate of mutant cells within the early lesions. Now they further checked how the role of the macrophages. They observed an increase in the proportion of the interstitial macrophages. Macrophages are basically one type of white blood cells and an important component of the innate immune system. They can tune the inflammation in our body. And they also notice that an increase in the PD-L1 expression on these cells in both control and engineered mice whenever they are exposed to the particles. And PM exposure is associated with enhanced and sustained long macrophage infiltration beyond the period of PM exposure. That means even at the 10th week. Then they check the effect of the early tumorigenesis. For that, they did the RNA-seq analysis. They had basically 4 sets of sample, tumor sample, lung exposed to PBS and PM, and tumor sample with the mutation exposed to PBS and PM. They observed that EGFR mutation is actually responsible for 38% variance of gene expression difference. They also get the late PM exposure led to an upregulation of genes which are known to regulate macrophage recruitment, including those that encode interleukin 1 beta, IL-1 beta, with abbreviated from. This IL-1 beta is basically a cytokine, and it has multiple roles in our body. It is produced by the activated macrophages. And it also mediates inflammatory responses. Then comes to the 8P2s. Then they concentrated on the 82 cells. These cells are in major lung epithelial cells as shown here. 82, 80 type 2 and 80 type 1 are there. They focused on 80 type 2 only. It behaves as a tissue stem cells that repair the epithelium during steady state replacement and after injury. And also, these 82 cells synthesize and secrete pulmonary surfactants, which regulates the surface tension alveoli and contributes to the host defenses. It is important because the surface tension actually controls the expansion and contraction of the lung. And it is also reported that the probable cell of origin of lung adenocarcinoma is 82 cell. That's why, because of its important role in case of the lung cancer, this isolated 82 cells from mice induced EGFR expression and exposed them again into PM or PBS. And they observed upregulation of genes previously associated on 82 progenitor cell states. An increase of 82 activated progenitor score is also observed when exposed to the particulates, both in the tumor sample and also tumor sample with mutations. That means EGFR mutated 82 cells are transcriptionally reprogrammed to the progenitor cell state following the PM exposure. Then the further explored the role of macrophages. PM exposed macrophages have shown enhanced organoid formation efficiency. That means PM-induced inflammation arises from the macrophages. And also they noticed that treatment of EGFR mutant 82 cells with IL-1 beta resulted in larger organoids. And these things can be prevented when it is treated with anti-IL-1 beta antibody. It shows the possibility of treating this type of cancers with anti-IL-1 beta antibody, as it attenuates the EGFR-driven lung cancer formation. So finally, it concludes that PM-exposed macrophages can induce a progenitor-like state in EGFR mutant 82 cells. Now the question remains whether these mutations are associated in the healthy lungs also. It is because this study is focusing the role of pollutants on the mutated role of pollutants in the lung cancer having EGFR mutations. Now, what is happening to the people whether if somebody is carrying EGFR mutation but not developed to lung cancer? They studied that almost 16% of people, although number is quite less, total number, but at least 16% of people are there who has EGFR mutations but never developed lung cancer. Now the question remains whether they are, how much is the possibility of having lung cancer in them in future upon exposure to the pollution. And this says that at least 1 in 554,000 lung cells would harbor an oncogenic mutation. They other checked for other mutations also, apart from L8058, they checked other mutations like exon 19 deletions and other point mutations which are known for the EGFR. And they checked the association of oncogenic mutations with non-cancerous tissue and exposure to the ambient pollution. Then they check the effect of oncogenic mutation in the healthy lung. And association of oncogenic mutation with the non-cancerous tissues and exposure to the ambient solution was observed. To explore it in a motor, they looked at the anthracosis. Anthracosis is a state of the lungs when it is basically pigmented or discolored, the lung. And it happens because of the accumulation of some pigments or some particulates, which very often is caused by the pollution, like exposure to the cooking fumes, soot, like that. Anthracosis is known to act as a surrogate marker of exposure to air pollution. And they observed an association between anthracosis and increased variant allele frequencies of EGFR mutations. That means EGFR mutations are related with the exposure to the pollutants. Although this research article is primarily focused on the EGFR mutations, but there are other genes which are also known to mutate in case of the lung cancer, like KRAS. And this research article shows that KRAS mutant clones may be more highly selected than EGFR mutant clones, and it requires a future studies. So the outcome of the research of this article is that PM can trigger the expansion of pre-existing mutant lung cells through an inflammatory axis, and temporal analysis suggests that 3 years of exposure may be sufficient to develop the EGFR-driven lung cancer. It provides a balanced study of cohorts. The cohorts includes wide variance in the sex, geographical origin, and ethnicity of the population. It reveals PM exposure contributes to the geographical disparities of EGFR-driven lung cancer, and we observed that PM induces an altered progenitor state in EGFR mutant 82 cells through the macrophage release of IL-1 beta, which promotes lung cancer. And this study comes also with a few limitations. One is that it is concentrated on the aggregated data. That means it looks only at the cohort data. It does not look at the individual patient. It does not look at the prognosis of the disease with the exposure of the mutant, exposure of the pollutants. That thing lacks here. And also, more work is needed to pinpoint which lineages harbor these mutations. And finally, this comprehensive analysis concludes that PM can trigger expansion of pre-existing mutant lung cells through an inflammatory axis, which can be treated by anti-IL-1 beta antibody. The temporal analysis shows that 3 years exposure process is sufficient to develop EGFR-driven lung adenocarcinoma, and it suggests a mechanistic and positive link between air pollutants and lung cancer. And finally, it provides a strong public health mandate to restrict particulate emission, specifically in the urban area. And the impact in the scientific context, the impact of the research is it shows another model to explain the lung cancer development. The well-known existing model is as shown in the schematic A here, that the pollutant mutagen molecules cause sequential accumulation of mutation, which leads to the cancer. But this study shows another model, which shows the pollutants promote the proliferation of lung cells that is already carrying pre-existing mutations. Here. That's why it shows somehow a warning to the people already carrying the EGFR mutations are more prone to lung cancer if they are living in a pollutant environment. And with that all from my side today, I thank everybody for your listening. If you have some questions, you can find me, you can write me at nupur@renix.org. Thank you very much.
Thank you, Nupur, for that informative presentation. Now the floor is open for audience. So Vijay is asking, was the particulate matter homogeneous in origin, or it's a mixture of different chemical entitles for those already known carcinogens?
No, it was actually mixture of different chemicals. It was not known to the carcinogens also. No.
Yeah, hope this answers your question, Vijay.
Yes.
Yeah. Further, I don't see any questions. So as there are no further questions, I think we can conclude the session. Nupur, please go to your last slide. So our next presentation is on 28 June. The presentation was given by Numer on the topic, an international multi-center matched cohort analysis of incidental meningioma progression during active surveillance or after stereotactic radiosurgery, the EMPASS study. Once again, I thank you one and all for your valuable time in spending for the CellTalk presentation. Thank you.